Picture this. Hi, my name is Dr. Harriet Kamendi and I am CEO of Kandi Bioscience. At Kandi, we help founders think through the strategies they need to bring their products to market. And in this video, I would like to describe the architecture that you need to build in order to take your product through the development phase. So on this slide, it depicts 5 rooms. And I like to think of my product development journey in terms of rooms. And in my rooms, the most important room is number 1. In that room, number 1, a lot of things are happening. You're operating in the pre-IND phase. The decisions you're making in this room are going to carry all the way to the end and beyond, actually, into the entire product development life cycle. And that means that the decisions you're making about the strategy, the population you're intending to treat, who you're going to test, all of these shape what your product looks like when it finally gains marketing authorization. And then when it enters the marketplace, all the decisions you made begin to unravel because all the things you failed to look for or looked for begin to play out in real time now. So there are 5 systemic failures that are fatal, in my opinion. And in the table below, we will assess the founder, action required, and then who is impacted and the consequences that each inherits. So the scoping the package for the IND reviewer only. So many people will develop a regulatory strategy that gets them to IND only. That is fatal. Why? Because if you only scope up to reviewer 1, then that means that you will be missing out on the other readers downstream of reviewer that need to read this assessment. So, for example, the first-in-human escalation lead of the pharmacologist, the carcinogenicity strategy team, the diligence toxicologist, if somebody's going to buy you, and then the marketing reviewer. All of them need to read this, the results of the IND, and then, of course, add the other required components. So every downstream reader inherits the package, and if you don't build for them, then you miss out answering questions that they are going to have. The second fatal failure is deferring metabolite characterization to phase 2. This is really serious, and the impacted reader is the phase 1 ADME reviewer, and all the other reviewers downstream of that. That's the carcinogenicity team, the diligence toxicologist, and the MAA reviewer. And this is because you always have to remember that metabolites differ between animals. Sometimes animals will have different metabolites than humans. And if you're not assessing the correct metabolites, then you will have to go back and conduct studies that you did not anticipate. The third fatal flaw is treating GLP as a status or a checklist rather than a specific decision. Studies conducted under strict GLP means that they carry quality assurance and they can be audited effectively. So when you're conducting these studies, you have to ensure that the company you're working with does not just have a GLP but they actually perform under GLP so that you avoid reworking all your studies and paying for unnecessary studies that you have to redo because the impacted readers do not trust the data you give them. The fourth systemic failure is not writing the first-in-human framework choice before the study design. So if you conduct your due diligence appropriately, then you should know that sometimes pushing the toxicology dose is not the only way of deriving the first-in-human dose, and that you may actually use your pharmacologically derived number to support your first in human dose. And if you did your literature review adequately, then this becomes evident. This is important because if you get to phase 1 and the first in human escalation lead rejects the NOEL, you have to do the MABLE. And that is a cost you did not anticipate or even include in your pricing. And the last but not least failure mode is the assuming CMC and non-clinical are separate work streams and do not need to run in parallel. No, they are conjoined twins and they have to run in parallel. And no matter how difficult it is to run them in parallel, you have to be thinking CMC before you dose. And that's because your drug substance in toxicology should not differ in impurity levels to your drug substance that was used to conduct your GLP studies. If they do differ, then that means that you're evaluating a product that you may have to reevaluate because you found out that you have new impurities that could be impacting the safety of the product you're developing. So, room 1, again. So as you conduct the work that needs to be done, you have to always remember that there are 3 parallel work streams that must deliberately run in parallel. They will intersect at different points, but they must run in parallel in order for you to ensure that you are anticipating the readers that will come downstream of 1. The first one is a non-clinical safety. The second one is PK, ADME, and TK. And the third one is CMC. So CMC is the drug product development. PK and ADME, that is just collecting blood samples from animals in every study, because you need to understand what happens when the drug reaches a certain threshold. And many people assume that you're going to have a general effect. Sometimes no. And therefore, you track PK to ensure that the effects you're seeing are PK mediated rather than anything else. PK or TK mediated. There could be other factors, other ADME factors that come to play, like absorption, distribution, metabolism, and even excretion. But you need to understand where they are changing. So it is important to track the life cycle of the drug in blood up to the end and in every study. So then the non-clinical safety are, of course, the studies that you will be conducting in animals. And many people will do this at specified doses. No, you're supposed to start by determining the maximum tolerated dose and then lowering that until you have a dose that enables you to assess risk for the required duration in the animal that you're testing. So before you start, here is a 90-day operation on a diligence baseline. The questions you must ask: who are the 7 readers of your non-clinical package and what does each need? For you as the founder, you're the operator. So this is your precise reader list and the routing map. And keep in mind that the non-clinical package actually runs from end to end. It runs from the pre-IND or the IND package all the way to the marketing authorization. And there are studies that must be conducted at each phase, and you need to understand which phase needs what study in place before you can go there. For the investor, all you have to do is wait for a Series A competence and discipline check. Second operational question that you must be thinking about is: what is your chronic dosing declaration under the M3 R2 Table 1? For the founder, here is what you need to be thinking: that your indication dictates how long your animal studies will be. And therefore, you must start with the end in mind. You must start with your customer, because you need to understand what your customer is and needs before you can even begin to design your chronic dosing study. For the question number 3, what is your framework choice for the first in human starting dose? You must understand, again, the founder, what what first-in-human dosing paradigm you're going to follow. Are you going to do an NOAEL, or are you going to do a MABEL? If you're going to do, depending on which you're going to do, then you need to be measuring or assessing certain parameters as you go along, because you cannot wait for the end. So for the investor, they need to look at the dose rationale memo, which is a high fidelity signal of program discipline. Then the fourth question is, what is your carcinogenicity assessment document? This document writes itself if you open the evidence log at the very beginning, at year 0. If you do not do that, then you will arrive at year 3 and may have to conduct an unnecessary carcinogenicity study. So a well-informed carcinogenicity study will be executed at around phase 2. And therefore, you have to have done all the other parts, the literature, the assessment of all your studies as you go through development. And last but not least, are your non-clinical and PK ADME and CMC work streams reading each other's work? Are they running in parallel? Are they talking regularly? They need to be loving on each other. So these 3 work streams, they are integrated, and they must be integrated in order for you to win. If you're only thinking about them separately, or they're not in the same room talking, then you're cooking a disaster. So the cost of missing room 1 has been already demonstrated by AstraZeneca in their 5R proof. And they showed that candidate selection nomination to phase 3 completion jumped from 4% to 19% just by building infrastructure strategy at year 0. So the cost to reach the proof of concept dropped by 31% and increased the enterprise asset value at years 3 and 6. So there is justification, or it's been demonstrated that if you do your infrastructure strategy from year 0, you will be able to gain massively as you go down the product development journey. So the other thing to keep in mind is that you always have to stay abreast on the upcoming guidances, because the ground is shifting very rapidly. So if you're not attuned to what is happening, you will miss and you will do unnecessary studies. So currently, the Senate passed the NAMS draft guidance, which was approved in March 2026, and the FDA Modernization Act 3.0, which was approved in December of 2025. And so going forward, there will be more utilization of in vitro systems and less utilization of animals only. And we can see this playing out in real time. Playing out in real time. So technically, if you want to build the universal package that can win, you have to think of the architecture very early. You have to start with your pre-IND package that anticipates the IND reviewer, the first-in-human escalation lead, the carcinogenicity strategy team, the partner due diligence, and the marketing reviewer that will come down the road. So you who's building a product, 2 directives you can execute tonight. 1 is the reader map. Write down the 7 readers your current package is actually being built for. If you cannot name them, no CRO will definitely be able to do it for you. And then the second thing is do a sanity check. Audit the intersection. And you want to force a meeting to confirm that your non-clinical PK and CMC leads are actively talking to each other and reading each other's summaries and memos, so that they can be able to ensure, so that you can be able to ensure that you do not repeat studies unnecessarily. Everything else follows from these 2 exercises. So in summary, safety is not a project, It is a promise that outlives the team. Most of the time, the team that writes the pre-IND package is not the team that gets the product to the market reviewer. So build it once, read it many times. If you need help evaluating your project, you can feel free to reach out to us. Our contact information is below. Thank you.